Hello everyone, my name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. In our last video, I have introduced SAMSON to you guys, and if you haven't watched that video where I have discussed from where you can download SAMSON, from where you can download the extensions, how much powerful it is, what is the expected futures, what I see for this program. And I also promised in that video that I will bring different tutorials for different tasks using Samsung. So today is the first tutorial of that series, and the tutorial name is how to perform docking or virtual screening with Samsung. From docking, I mean docking of a single compound or an individual ligand. But if you have more than one ligand, let's say 100, 200, 10, 5, 6, whatever, that depends, then we call it virtual screening. So how we can perform virtual screening using Samsung or Autodock Vina. It is an extension. Its name is Autodock Vina Extended. Uh, the extension is included in Samsung. So let's begin how we can do this kind of docking and analysis. In today's tutorial, I will use 4075, which is the bromo domain of human BRD4 in complex with fostamatinib. This is the structure I have already downloaded from the PDB. When you open PDB, your structure in PDB, you can click here. I can show you there, no problem. Okay. And for ligands, okay, the spell is wrong. Pardon. Okay, for ligand, we will use PubChem and we will download randomly 4 to 5 ligands, and then we will dock in today's tutorial those ligand, and will perform some preliminary analysis using Samsung. So let's start. Okay, here is the PDB page, and if you don't know how to download the structure, just click here, download files, and download the PDB format. I have already downloaded. The structure is present here 4075 in my PDB in my download directory. Okay, we will also use PubChem. So let me open it in advance. This is a PubChem. We will download randomly 4 to 5 ligands, and we will test it there against the bromo domain of human. So let's start. So first of all, we will need to create a separate directory. Let's say I'm creating this directory in the download folder, and I will keep its name docking with Samsung, docking underscore Samsung. Okay. Then you can cut this and make another directory here, receptor or whatever you call it, control V. You can also make another directory. Its name will be ligands or ligand library. It's up to you. And another library, we will call it results. Okay. In the receptor folder, I dropped the protein structure we will use. In the ligands, we will download some ligand from the PubChem and we'll keep it here. And in the result section, the result directory, the program will download or will place some results here, and we can analyze that result later on also. Okay, so we have a receptor now. I will download randomly some receptor from the PubChem. Let's say I download ibuprofen. Oh, let me tell you that I'm downloading these compounds just randomly. No literature study, just because my main purpose is to show you how we can perform docking using Samsung. So this is a 3D conformer. Download coordinates and save the SDF. Okay. Now download some another compound. Let's say uh, AC, aceto, acetochlor. Okay, acetochlor. Once again, I don't know about anything about this compound, just a random compound. Let's see. 3D conformer. This is okay. Download. Okay, third, I'm going to download some antibiotic. It's okay. No, I cannot see any structure for it. So let's say again, I can see literature. Oh my God. Let me try this one. Acetaminophen, like here, generally we call it paracetamol or Tylenol in some countries. Okay, download 3D conformer. So this is the third structure. Let me download imatinib. Say, okay, so these structures should be present, need to be present in the download directory. Okay, they are here. Let me cut it and put it into the ligands folder. Okay, to keep things clear, we have a receptor, this one, or protein, and also let me tell you, the Samsung can also handle more than one receptor, but right now we are going with a single receptor. And this is our list of compounds, 1, 2, 3, 4. We are going to test them. And if you want, you can rename it here. Otherwise, don't waste our time. I'm not going to waste time on the renaming. So this is our compound library, and we will ask the program to place our results here. Okay, so that's it. Now we are going to open Samsung. I will try not to discuss the other features of Samsung, and in today's video, I stick myself only how we can perform the docking job. Okay, this is our Samsung, and how we can open, first we need to open the receptor. Click here, the file menu, then open, then In the download section, this is our download directory. We have a docking Samsung, and we have a receptor. So I open it. Okay, this is our receptor file, and it already contains uh, some another ligand, co-crystallized ligand, and its name is, let me, this one, fostamatinib. Okay, whatever. Okay, if your ligand does not contain, if your receptor does not contain any ligand, then also okay, we will discuss how can you go in that scenario. Also, let me tell you that the Samsung program can perform covalent docking, normal docking, flexible docking, rigid docking, whatever you call it. So let's proceed. Okay, now you can see home. In the home tab, you will see app. Click here. I have already the Autodock Vina extended, but if you don't have, click here and then click add app. From the app section, you can sign in. Already, I have covered this thing in my first video, and click here the free trial. Let me show you again. And then once you are signed in, because this time I'm not, then you will see the add button. Just click here and restart your Samsung. Then you will see this kind of extension here in the apps. So just click here simply, Autodock Vina extended, and you will see this kind of thing. Now, if you want to resume the previous project, if you have already uploaded results, if you have performed docking on Samsung the other day, then you can load those results directly here. But now we are going to click new docking project. Click here. Now, first we want to prepare the system. The beauty of Samsung is that you will not go to anywhere else, and it's very easy. So how to prepare the system? Let's see. Here is the home. In home tab, you can see prepare. Click this. Now it gives you 5 options. Remove alternate location. It's better to turn it on. Remove ligands. Remove water. Remove monoatomic ions, and add hydrogen. So currently I want to remove water. I will add hydrogen, and this one will also be turned on. Remove alternate locations. Okay. Remove ligands. If you want to remove ligand, then this ligand will be removed. But in that case, I will suggest you to save your ligand and upload it here separately. Right now, I don't want to remove it from here. You will see how then I will proceed. So you will see. And remove monoatomic ions. So far, I know I cannot see any monoatomic ion. The red color, these are water, and this one is that fostamatinib, I think. Yes. So no monoatomic ion, so it doesn't matter whether you turn it on or turn it off. So just click OK. Now the system is prepared. You can see, let me drag it down. The system is prepared. This is the co-crystallized ligand. Okay. Now, this is the hierarchy window. If you have used already like the Discovery Studio Visualizer, they are also following the same kind of hierarchy like Samsung. Okay. This is our receptor file 4075, and the presentation is ribbon. If you want to turn it off, click it here, otherwise leave as it is. Now we want to set the receptor. So once you click it set, it will give you like please select the receptor. For example, because the program don't know which one is receptor. So how to do it? Click here. Once you click here, if you can see, the whole receptor is selected. Also the ligand is selected. So we want to exclude this ligand, but how we will do it? Just see. This is the selection tab. Click here. Now we will filter the selection with the select ligand. Okay. Now when we select this part, it will only select the ligand. And you see? Okay. Now the current selection is 2RC. This is the ligand name. This is the index number 201. Right click and then say extract structure. And write it here. I say 2RC. 2RC. Oh, wait. 2RC. Okay. Now the structure is extracted. If you want to see how, let's see. This is the receptor. You will not see the structure here. You see, it's finished. We have removed already the water, so that's okay. And the ligand is here. This is the ligand structure. Now, if you select this receptor, it will not select the ligand. Can you see? So isn't a great function? Okay. Now click here, set. So you will see this kind of box that covers all the receptor protein. Okay. So now the receptor is set. Another beautiful thing, which is not as simple in the if you are using Autodock Vina with the MGL tool or the other softwares, flexible side chain. If you want to perform flexible docking, and if you want to perform, let's say, if you want to perform this area, this side chain, a flexible one, you can, by the way, click here and deselect because the program has already accepted it. So if you want to use this area as a flexible because it is a loop, then you can select this area from this menu. That is not so difficult. From this selection filter, just come here, let's say residue, and now you can select it. You can see. So once you click here, it will be selected also ASP88. This is Feline 87. You can see like this. So you can select this and then after the selection, click here. So the program will deal this side chain flexible. And if it is required to provide a conformational space to your ligand, the program will provide it. So we call it a kind of flexible bond. But remember, flexible side chains. But remember, uh, it will increase your docking time. How much? That depends on the protein and the structure of your ligands. Okay. Rotatable bonds, if you want to make some bonds rotatable, so you can do it, but the rotatable bonds will be turn it on if you are using this selection, this side chain selection, flexible side chain selection. I'm not using it, so that's okay. Now, the second window is Set Ligand. This is an important window. I know most of you will ask me question in comments that my ligand do not contain any, my receptor do not contain any co-crystallized ligand. So what to do? So let me tell you. Um, from here, first we have to select the ligand library. What you will we will select if you are using a single ligand, if you have single compound, just click single document and then set that ligand, load that ligand here, then click it, select it just like this. Select it and then click set. But if you have more than one compound, like in the case of virtual screening, so we will select ligand library. In this case, we are using a library of ligands that contain four compounds. Initially, we have downloaded from PubChem and they are placed here. 1, 2, 3, 4. So we have this ligand library. So select this option ligand library. Now, where is the ligand library? Click here. Then come to your download and the docking Samson. And this is our ligand library. Its name is ligands and four structure are there. You cannot see that's okay, but program will see it. So we have provided root or path to the ligand library. That's okay. Now, if you want another beautiful step or a greater, much appreciated one, if you want to perform minimization on your ligand library, then you can take this on uh, otherwise you can turn it off. You can choose different steps 10,000, 5,000. By default you will see 2,500 I think, but I have set it for myself or for this. So to be a quicker one, let's set it to 2,500 step. Now also click here, add missing hydrogen. Okay. And if you want to choose some rotatable bonds, then click here. But if you don't want, then leave as it is. Okay. The third, we have choose our ligand library. We have selected our receptor. The third menu is about the grid. Grid maps, a selection of grid maps. The grid maps mean where you want to dock your ligand. Okay. Now, in this case, if I have this ligand, so I will use this region. But if you don't have any ligand here in the PDB structure or in your model, then you just need to click this receptor. So it will select the whole protein structure and will perform docking to the pocket. Okay. And then you can confirm from the literature study whether the docking is correct or not. Okay. Now come here. I will select pocket selection. But how should we know that where is the pocket? Then Samsung provide another interesting feature. Just click here, the select button and click biology. Now, the second tab is binding site. So click here, binding site. You will see these options, ligands, auto detect ligands, Yes, that one is okay because the ligand is present here, so the program will detect it. Then we ask like around this ligand, use 5 angstrom region and atoms and bonds, and this will be our binding pocket. If you want to increase this area, so click here, it will be 6, 7, or if you want to decrease, that is up to you. But by default, you will see 5 angstrom. So just click here. Once you click it, so you can see some sideways pockets are selected. Let me zoom it. Can you see here? These regions are selected or the 5 angstrom um, around this ligand. So now pocket selection. Okay, click set. Oh, I selected. Okay, let me do it again because it's my mistake. Okay, do it this auto detect ligands. This wait because the use original auto detect ligand. Okay, because the ligand we have removed from here. That is the thing. So that's why it is not able to detect uh, that ligand. Okay, now it has detected. Okay, but another beauty, if still you think that this place is not sufficient enough, then you can keep here and drag this box. It's very easy and very interactive. You can see. Okay. So this is our, how to say, this is our grid location. Let me, if I want to make it bigger this way, okay, okay. So now once your grid is ready, this is our grid set. I have already clicked set. Now we have few more options in the stage 4. One is score only, local optimization only, or generate PDBQT files only. I will choose dog. That is okay. Now click here, this option, and come here. Samsung supports two programs, Vinardo and Vina. But we are preparing our system in the Vina, so we have to select this Vina. Now, if you think that your structure are bigger one or complex one, or if you think for a higher accuracy, you are planning higher accuracy, then I would suggest to make this exhaustiveness for global search double, let's say 16. But I will not use 16 because it will multiply the docking time. So my main aim is to give you a demonstration and uh, teach you how you can tweak things, different things. Okay. Maximum number of generated binding modes. Normally, if you are using Vina with other software or with other tools, so Vina generates 9 binding modes. But if you want to generate more than 9, so just increase it, or if you want to generate less than 9, so you can decrease it. Okay. If you want to include Monte Carlo simulations, um, for example, what are Monte Carlo simulations? So, in short, if your binding mode is selected, let's say the best binding mode is generated, then the program will simulate it, that binding mode, like, and you will have much more higher accuracy. But remember, It will take uh, much more time also. Okay, minimum RMSD between output poses. That's okay. Energy range like this. It means that if uh, if the energy is above going to reaching above this, docking will be stopped. And limit the total number of modes in the memory. For example, if you are using a system with a low RAM, RAM, then I would suggest to reduce this number. Otherwise, you will feel the system is lagging. It's okay. So now click here, and if you want to include some extra weights from other programs or from other calculation, you can turn it on and include it. Okay. So finally, do you want to save your result? Yes. So just click here, and we have the directory already created. That is the results directory. Click and choose. So the results will be saved there. Save receptor in file with resulting ligand mode. Yes. Okay, now we are going to dock the library. So just a quick overview. Here we have selected the ligands. Here we have selected the ligand library. We have also selected like each compound should be minimized for 2500 steps. You can increase or decrease it. This is the grid setting. We set it based on the pocket selection. This is the docking. Here you can change different options like exhaustiveness and generation modes. This is the results we are to save. This is the dock library. We will need to click and it will start working. So personally, I also don't know how much time it will take because I downloaded these compounds randomly, and for random compounds, you cannot say. But I believe it will take less than 5 minutes. So let's see. It's still running. You can see 75%. You will see here. Okay. Not to waste your time, I'm going to pause this recording. Okay, welcome back. I got this menu on the screen. The docking has finished. Would you like to load the results? And believe me, it took almost 2 or 2.5 minutes. Okay, yes, I want to load it now. Yes. Now, okay, they're asking you how many or what you want to load in the menu. So, if you select it to download all the results, you will see a long list here. But actually, I want to load only the top molecules from the selection. Okay, uh, when the docking finished, so I forgot to turn it on the resume button. So sorry for that, but you will see a kind of window here, and the window will ask you that top how many poses for top ligands you are expecting. So I choose one for each ligand in the second row in the second menu. But if you are using Samsung for the first time, I will ask you to choose all the nine. Then you will have this plot, nine here, nine for this compound, nine for this compound, and nine for this compound. Okay. But why I choose one? because I wanted to, I only selected the top compounds from each section. Okay, once you do that, you will see this kind of figure here, like this. Here you will see the different scores for this conformer or for this ligand, that is -8.322, and for this -6, and for this -5 and -5. Okay, as we did not rename the ligand, so therefore we only know from the number like 5291 is the best. So if we see here, the 5291 is the last compound we downloaded, and that was, I think, imatinib. So it means imatinib is the best among all. Okay. Now you will also see this window here. You can drag it here, and it will give you a very beautiful image. You can also export this result in the CSV file, export table. Let's say export table. Then I will try to save it here. Let's say res1 or result underscore one, and just click save. Okay, you will see here result 1. Now you can see the conformer 1, this index name, these are the ligand names, and -8. These are actually the top from the pool because we have generated 9 for each ligand. -6, -5, and -5. Okay, now what about this figure? Can you save it? Yes, you can save it. You can also export this figure. So save image. Let's say I name it figure one underscore one, and open. Here is your figure. Now, if you see, we had only four compound, one, two, three, four, and I selected only one, the top one from each. So if you are doing this for the first time, because I did this before also, so you need to select all the nine. So you will see the difference, the distribution difference in this compound, and in this, in this, in this. Okay. So this, you can use this plot. Now, once you load the results, you will see this kind of thing. So why you see a kind of ball and stick representation or leg query representation, also the ribbon, because this they have loaded the other receptor, so you can turn it off by clicking this. Now, the second thing you need to turn it off is the, how to say, this box if you don't like it. But just come here. You can see dock ligand. So let me turn it off. This is the original ligand. Click here. Now it's disappeared. Now let's see this one. So this one is here, very deep. Okay, you can see this one. This one is here. Now the third one here. And how about the fourth one? It's here, the one that scored highest. Okay, now if you see, you will see four tabs here. So now we are in result tab. If you click here, log. So you can save this log with yourself. This is the docking 9 pose and their energy for this compound, the first one, and then for the second one, for the third and fourth. Okay. Now, how to uh, do it this way? Wait. Okay. This is the result. Now, how to we are going to disappear this again, the this box. Click here, dock, and just click here, show box. So now you will not see that box thing. That's it. Now simply we want to analyze the interaction panel. So first we will use this conformer or this compound. We have all four. So this one is selected. So let's see. This conformer is selected and we want to see its interaction panel. So how to do it? Once again comes here to apps. You have two apps here. One is hydrogen bond finder, another one protein ligand interaction analyzer. Okay, so we will use the first one. I have already this thing. First, we will try protein ligand interaction analyzer. Click here. So, once you click here, it will ask you which one is the receptor. So, if you remember in the first step, we have removed the ligand, the co-crystallized ligand, and it is only the receptors. Click here. Uh, wait. Oh, it's here. Okay. Click here and then set. The receptor has been set successfully. Now, a single ligand, I'm going to test it on this one. So we just select this one. Let's say this one. Okay, now you can see it has been selected. So set. Now we have different options. Compute SASA for solvent accessible surface area. And this is the SASA radius. This is the surrounding residue cutoff. 5 angstrom for the hydrogen bonding. The cutoff distance is 3.5. If you want to increase it, let's say you can do this way. And these are the acceptable angles for the hydrogen bonds, and the donor receptors are explained here. Just click analyze, and we see here we have four how to say hydrogen bonds. You can export it in CSV. Let's say protein PL interaction analyzer. So save. We have this result now, and we can open it. So it says that four hydrogen bonds and the molecular weights and some receptor and blah blah blah. Okay. Now, if we want to see our to see these bonds, we can use another app like this one, hydrogen bond finder. You can, and it's almost a similar one. Just add hydrogen bond to the visual modes. Okay, so between receptor and ligand, so what we do first, come here, click this receptor, then press the control button and select this. So both are selected, and let's see the visual mode. Okay. Uh, okay. So we have, oh my God. So actually, okay, it's fine. So the moods are on. You can see, wait, you can see here, this is the hydrogen bond. But why we cannot see the other thing? Because this is the ribbon diagram. So for that, that is very easy. Select those residues which are forming the visual modes and make it into the ball and stick. I'm going to pause this video or to close this video because it almost 50 minutes. So by this way, you can calculate. Okay, one question you can ask or you may have in your mind that um, the thing, the this, it shows number of hydrogen bond one, that shows number of hydrogen bond how to say four, because there we have changed the distance from 3.5 to 4, and if you change these distances, tweak these distances, you will see four hydrogen bond also here. So nothing a big difference. And wait, let me try to show you some one last thing. Home, edit and select then biology and then this and then this and then group one and then yeah, I was trying to generate label but it didn't work. It's okay. So overall, that was the docking. How can you perform docking? I tried my best to explain each and everything for you, and the results are already here and should be here. Yes, you can see this is the result section. PDBQT files, receptor, ligand, same. Ligand same means the ligand with uh, the receptor. Sorry, without receptor, but that is easy. You can open this with the receptor. Um, you will be able, you can also save a PDB if you want. Otherwise, you may not need that. The same mean the Samsung file. Uh, so this was all about the docking. You can perform docking here, and at sometime we will also cover the visualization thing. Then I will show you how to generate some beautiful figures. If you want to generate simple figure, just click here and it will be saved. And the figure is saved, let's say, in documents, one in strong and capture section. So, but I'm not sure because these will be it will not be a high DPI figure, this one. Okay. So you can, if you want to change the background, you can change the background with this button. I have covered these things in my first video, and I hope I have covered the main theme of this video. So if you're liking my videos, please subscribe and support my content so that I can bring much more content for you. Okay, thank you so much. Bye bye.